Hi, everyone. From the Scientists in Every Florida School Initiative, a program led by the UF Thompson Earth System Institute, I am Brian Abramowitz, and along with my colleague Stephanie Killingsworth, we would like to welcome you to our second Nature of Science webinar. Our mission at the Scientists in Every Florida School Initiative is to build long-term collaborative relationships between teachers and scientists to better integrate current scientific research and big data into classroom lessons that adhere to Florida sunshine standards, and in doing so, to connect the scientists with every Florida school in the state. Today, we are joined by Dr. Emily Durkin. She received her PhD from the University of Alberta in Canada in biology and ecology. As an evolutionary ecologist, she spent much of her time studying the relationship between parasitic lifestyles and their hosts. She's here today to talk about some of the research that she's done and it's relevant to your classrooms. I would like to introduce and hand things over to Dr. Duncan.
All right, thank you very much. I wasn't expecting an introduction, so I have a couple of introduction slides. So let's see here. Okay. Okay, can everyone see my screen and the PowerPoint?
Yes.
Okay, excellent. So a little bit more about me. So I'm originally from Madison, Wisconsin, and I did both my undergrad and my master's work at Northern Michigan University by Lake Superior, and I studied chewing lice on brown-headed cowbirds. And if you know much about birds, brown-headed cowbirds are actually parasites themselves. So they lay their nests in the eggs, or they lay their eggs in the nests of other birds. And so they had the potential of maybe having some interesting parasite assemblages on their bodies, but I found that they actually didn't have very interesting parasite assemblages on their bodies. So that was kind of cool. It's how science goes sometimes. And then, as Brian had mentioned, I did my PhD at the University of Alberta in Edmonton, Alberta, Canada. And there I studied specifically a fly and mite system to learn more about how parasitic strategies evolve in free living lineages of animals. And so currently I'm a postdoc at the University of Florida in Nick Kaiser's lab. He has a behavioral disease ecology lab. And so I've been focusing mostly on studying the attachment behavior of these mites, so actually trying to quantify it. And so currently we have a paper in review about parasitic personalities in these mites. So we found that some mites are really interested in always attaching to a host when they get the opportunity, and there are other individuals who never want to attach ever. So it's kind of cool that we're seeing that there are inherently more parasitic mites and inherently less parasitic mites, which is very cool. So today I wanted to talk about evolution and an evolution experiment that I ran during my PhD. And so first, I did want to just propose a question. I wanted to ask, what is evolution? And we can poll everyone. So it's a little complicated, but it will get better. I promise. So I'll give like a minute or two to read through the answers.
So far, we have 2 responses saying the second option, change in the characteristics of a species over several generations and relies on the process of
Natural selection.
Yeah.
Shut up.
We're up to 3.
Okay, so is that what we have, those 2 answers?
More and more people are saying that same response. But we got a different
Okay.
one. We have one person said change in the heritable characteristics of a population over successive generations.
Okay.
And now we have one for a process of gradual changes that take place over many generations.
Excellent. So it looks like, so no one picked the first answer?
No one.
Okay. So the reason I chose to, I guess, ask this question and given these particular answers is because evolution, I think, is a confusing subject. And it's difficult to know what it is because there are so many different answers that are available to us. So every single one of those answers I actually pulled off of a Google search for evolution. So I just typed in evolution, and these were all definitions that came up, I think, on like at least the first page. And there's only one answer on there that I like in particular that I think is actually correct. And the others aren't so great. And I'm going to walk through right now just so that everyone can get a better idea as to what evolution actually is. So the answer I like most is what was C, the change in the heritable characteristics of populations over successive generations. And that's because essentially evolution is a change in traits within a population over time. So in this picture, oops, I have a population of flowers and the trait difference is color. And over time, if there's a change in...
I don't know this already.
Then it's, that is evolution. Now, A, the gradual development of something, especially from a simple to a complex form. So I don't like this answer because you don't necessarily have to evolve to become more complex. So in the case of tapeworms and lots of other parasites that evolve from free living ancestors, they become actually a little more simplified. So in this worm here, it has eye spots, it has a nervous system, it has a GI system, and tapeworms, which are the more evolved parasitic form, actually has completely lost a lot of those body systems and it doesn't have eyes anymore. So evolution does not necessarily mean you're becoming more complex. B, change in the characteristics of a species over several generations and relies on the process of natural selection. So the reason why that answer I did not like is because evolution can actually occur by multiple processes. So natural selection is one process by which populations can evolve over time, but there are other forms of evolution. So on the left here, I have genetic drift, which is, it kind of describes random evolution. So in this cool example, you have a population of insects with a different ratio of red and blue insects. But in a random event, like some of them getting swatted, you now have a change in the number of traits being expressed within that population, and that is evolution. So evolution can be completely random through genetic drift. And also on the right, an example we're all pretty familiar with is artificial selection, in which pretty much only humans choose the traits that they are interested in, and they selectively breed those traits. And that's what we've done with dogs. So within a single animal species, we've been able to evolve all of these different breeds expressing the traits that we are interested in. The last answer, a process of gradual changes that takes place over many generations. So the reason I wasn't as excited about this answer either is sometimes evolution can be quite fast. It doesn't have to be really slow. So one example is through antibiotic resistance with bacteria. That can happen quite quickly. And another really cool recent study, a researcher was on an island and he was measuring the morphometrics, so leg length, and just all sorts of anatomical features of some animals. And right after he made his initial measurements on these animals, 2 hurricanes, Irma and Maria, swept through. And he took this as an opportunity to go back and measure the traits again in these animal populations. And he found that just after those 2 hurricanes, he found a decrease in limb length, which helped them be less likely to get caught up in the winds. And they also had an increased toe pad size to help them hang on during the hurricane. So there are many instances in which evolution can occur quite quickly as well. It doesn't have to always be a really slow process. So again, evolution is just the change in frequency of heritable traits within a population. So getting into my work, so I wanted to walk a little bit through experimental design somewhat. So the question I asked was, how do parasites evolve from free living ancestors? And my hypothesis, and the hypothesis proposed by other researchers, is that foracy, and foracy as a definition, foracy is when smaller organisms attach to larger organisms as a means of dispersal. So I have this picture here, there's a little pseudoscorpion attached to this fly here to help them get to places that are too far away for them to travel to on their own. So the hypothesis is that this phoretic relationship may be served as an evolutionary stepping stone or a gateway towards obligate parasitism, such as seen in this tick. So maybe over evolutionary time, ticks were simply hitching a ride on animals and then eventually began feeding on the animals they were just hitching a ride on. So for this hypothesis, I thought, if this hypothesis were true, that foracy served as an evolutionary stepping stone towards parasitism, I predicted that host attachment, so the act of actually hitching onto a host, must be heritable and capable of evolution. And so I decided to test this hypothesis using artificial selection. So in order to artificially evolve an animal or a population of animals for a trait of interest, you need a species that exhibits 2 things. And in my case, my trait of interest was host attachment. So I needed a population of animals that exhibits host attachment for me to select upon. But also, very importantly, they had to show variation in host attachment, because if all of the animals within the population exhibit my trait of interest, I can't really change its frequency in the population because 100% of the population is expressing it. So I worked with what's called a facultatively parasitic mite, which simply means they can or cannot exhibit parasitic behavior, and they don't need to exhibit parasitic behavior in order to survive and reproduce. And so the mite was Macrocheles muscodomesticae. This mite is found right here in Florida. So here I have a little video. This is a mite crawling around in just some rotting organic matter. So they're commonly found in compost as well as dung. And sometimes these mites, when given the opportunity, will attach to a fly. So here I have a mite attached to a fly. So this was a perfect study system because some mites do attach to flies, others do not attach to flies. So I had lots of variation. So next, in my experiment, I needed to be able to identify the individuals that expressed my trait of interest. So what I did, and I have another video here, so the little red-brown spot in this container is the mite, and here's the fly. And so I exposed mites, so lots of individual mites, I exposed them to a fly host for about 1 hour. And then after that hour, I scored every single mite as either attached or unattached to their host. And then I can select those individuals that I was interested in to produce the next generation. So in this case, I was selecting the mites that had attached to their fly and placing them into media so that they can reproduce and produce another generation. And this was something that I could then do for multiple generations over and over again in order to see if I can get a response in that trait. So I couldn't just stop there. I wanted to perform rigorous science, and rigorous science is defined as theoretical or experimental approaches undertaken in a way that enhances the confidence in the accuracy of findings. So I couldn't just select for the trait in one population of mice. There were some other things I had to do in order to make sure I was truly seeing an effect of my selection. So I had a control group, and control groups are really important. They're maintained under the exact same conditions as the experimental groups, but they don't receive the experimental treatment. And so that makes them as a good benchmark for us to compare our experimental treatment groups to see if we actually see an important and meaningful difference between the two groups, which would imply a potential effect of our treatment. So in my selection experiment, I exposed a bunch of mites to flies, just like I did in my experimental group. But instead of selecting the mites, only the mites that attach to the flies, I randomly selected mites regardless of whether they attached to a fly host or not. And this served as my control group because they weren't receiving the treatment of my artificial selection. And then I could compare what I saw in my selected group to my control group to get a much more meaningful comparison to see how much of an effect my artificial selection had on the mites. And then lastly, replication is incredibly important. So as I talked about earlier, evolution can occur completely randomly due to genetic drift. So if I had only done this in one population, there's the possibility that the results I saw were just a random act. And I wouldn't know that that was a random act unless I repeated this experiment over and over and over again. And if I continue to get similar results, it makes me much more confident in the results that I am obtaining in my experiment. Whereas if I only perform it one time, I have no idea if that was just a fluke occurrence that one time, or if I'm truly seeing a biologically relevant effect. So I did run this experiment 4 times. I wish I could do more. 4 typically isn't a great number of replications, but given the logistic constraints and how difficult it was artificially selecting mites, I think 4 was pretty good. So on this graph, I have generation on the x-axis, so basically time. And on the y-axis, I have the proportion of mites within the population that attached to a fly. And you can see after about 5 generations, we start getting what would be considered some significant divergence. So where there is a significant difference in the attachment prevalence between my selected and control populations. And if I measure just divergence, which is the difference between the selected and the control populations over time, you can see there's a positive relationship between the differences between the selected and control groups over time. And from that measure, you can actually calculate heritability, and it's also a really good indication that, yes, this trait is heritable, which again, as a reminder, was host attachment behavior. And it is evolvable because I indeed was able to experimentally evolve it within this population. And because I was rigorous, which is really important, I was able to publish these results in a peer-reviewed scientific journal, which is always very important. So my conclusions, my prediction that the host attachment trait is heritable and capable of evolution, my data supported this prediction, which is awesome. Which in turn supports my hypothesis that 4C served as an evolutionary stepping stone towards parasitism. However, I do want to make sure to point out that although my data support this hypothesis, it doesn't prove my hypothesis true. There still could be lots of other hypotheses out there that my data would support. So at least in the scientific world and using scientific language, we can never actually prove a hypothesis to be true. We can only reject and prove a hypothesis wrong. So as of now, my data do support this hypothesis, but it doesn't prove that hypothesis. And then lastly, I just wanted to mention that experiments are really messy. Whenever I'm presenting these data and talking about these experiments in order to make things quick and easier to digest, I don't go into all of the issues with trying to set up an experiment like this. So I just want to keep in mind that experiments are messy and never really go as well as planned all the time, and that is totally okay. All right, so thank you for your attention, and I'm happy at this point in time to take any possible questions.
Okay, so chat box here, if you all have questions, you can type them in the chat box, and can everybody hear me?
Yes.
Okay. I also, while people might be typing right now, have a list of some nature of science standards that I thought would be nice just to share, really to jog memory and make you think of ways you can use this. In fact, Emily, I thought what you said was so profound and such a great example to share in the classroom, especially for a lot of the middle and high school level. It's a perfect example of the scientific process for them to see with research that you've actually completed. I think it was a really good piece just to even share for a 15 minutes class exercise. Looking for questions.
Bruce, why don't you start us off?
Yeah, I thought this was a fascinating talk. Thank you so much. Really interesting. I'm fascinated with the this photograph. I mean, how many mites can a fly? What's the load there in terms of the cost of carrying these mites around? That must be really quite significant. How many typically in the wild? How many of you? What's the maximum amount of mites you've seen a fly carry? And the other thing I have is, are the mites species specific to the flies? So do they care what species of fly they're attaching to? Is it only one species, or are there multiple species that they might attach to?
Excellent questions. So first, with how many mites do I typically see on a fly? In the wild, I usually find, so when I go out to actually collect these mites, I go to compost piles and I use a net and just catch a bunch of flies flying around, buzzing around the compost. And so I usually see, I think I've seen maybe up to 5 mites on a fly in nature. In the lab, when you can observe the mites doing whatever they want to be doing, and the fly doesn't necessarily need to be able to fly. I've seen with this species, which is a much larger species than other species, I've seen like the body completely covered, so up to like 20 mites. And with smaller mites, I've seen like up to 50 mites on a single fly, which is pretty amazing. But in those cases, the fly isn't able to fly. So it probably is happening in nature, but I'm not able to see it with the way that I collect the flies, since I'm only selecting flies that are able to fly. Second, what is, because I think I heard kind of a question of what is the cost to the fly. So in one of the labs I worked in, they studied the fly more so, and we found that the flies actually have lower reproduction. They have typically shorter lifespans. They're not able to fly as far or as long as flies without mites attached. And it's kind of interesting. We saw the same results even if you take the mites off. So obviously there is a physical restraint with having such a large and heavy animal hanging onto you, but even if you take the mite off, you will see those physiological costs still manifesting, which is really cool. Lastly, are mites species specific? These mites are not. So you can actually find this species of mite on any insect, and they have been found on multiple insect species, but they are, I think they prefer flies, but they're not specific to a fly species. We use Drosophila in the lab just because the fruit flies are an easy system to maintain in the lab.
Thank you.
Yeah, of course.
Hi, this is Christy from Seminole County.
Hi.
I'm still learning how to work this chat, and I typed it just to you. Sorry, I would love for the whole group to hear it.
Oh, I don't know. Did I see it? I don't know if I don't.
It's okay. I'm going to ask it out loud. So
okay, great.
Thanks, Christy.
I'm in Central Florida, and I'm also a beekeeper. And being a beekeeper, I'm no stranger to the Varroa mite. Are there any closely relationships from the Varroa to the mites you're talking about?
Yes. So, mite taxonomy and systematics is, I think, relative to a lot of other animal groups, is really messy because mites are incredibly speciose and numerous, and not that many people study mites, so it means their systematics are a mess. But if I remember correctly, the entire group of mites is split into two smaller subgroups, which are the Acariformes, which are called the mite-like mites, and the Parasitiformes. So these are the more parasite or parasitic-like mites. And both Varroa and this mite, as well as its many relatives, are both members of the Parasitiformes group. Oh, and also, maybe of interest, ticks are also members of this group. So for those of you unaware, ticks are mites, and the tick family, the group of ticks, fits within this Parasitiformes group of mites, along with the Varroa mite, as well as this mite Macrocheles.
Great, thank you.
Mm-hmm.
Do we have any more questions out there? All right, as promised, I'll just share this. Oh, I cannot share screen. Hold on one second. Emily, you might have to unshare yours for a second.
Oh, okay. Stop share.
Okay, let's see if I can share the screen. So this is just a conglomeration of nature of science standards, again, of which I really feel you've hit a bunch of. Also, there are several standards for middle and high school, comparing and contrasting relationships among organisms, particularly the symbiotic relationships, in this case, parasitism. So those standards would be really nice to maybe circle back and bring this conversation up in class when the time is right. And again, infusing experimental design throughout the year, this is a great anecdotal case to share with your classroom. And I think with the videos and the slides, it's very easy to see some of those steps. So any questions out there?
Thank you for digging down all the way to 4th grade. That's awesome.
Absolutely. Well, we definitely want to hit the broad spectrum. So
Yes, thank you.
sure.
Hi, Emily, Doug asked the question, do mites hook onto other mites?
Oh, excellent. Yes, sometimes. They are very happy to eat each other, like many invertebrates. But I have seen a few cases of, so again, like the diversity of mites is ridiculous. And there are some mites that are quite large, 4 mites, and then there are other mites that are incredibly tiny. And I have collected, and I've had others bring me mites with different mite species smaller than them that are attached to them, which is really cool. I'm not sure whether the relationship is parasitic or simply phoretic, like I'd already explained, but it is very cool. Yes, mites will attach to other mites.
Awesome.
Everything's got to eat.
That's right.
Right.
That's right.
I'm gonna pop that guy off so I can see your face again. All right, so I thank everyone very much for being on the call. Emily, we really appreciate everything you do, and thanks so much for sharing and communicating your research with us.
Thank you.
Oh, thank you. Thanks for having me. I love doing this.
It was really good. Thank you. Very fascinating. Thank you so much.
You're welcome.
Have
All
a good
right.
evening.
Good night, everybody.
Take care.
Bye.